zDNA is one of the many possible double helical structures of DNA. It is a left-handed double helical structure in which the helix winds to the left in a zigzag pattern, instead of to the right, like the more common bDNA form. zDNA is thought to be one of three biologically active double helical structures along with aDNA and bDNA. zDNA is quite different from the right-handed forms. In fact, zDNA is often compared against bDNA in order to illustrate the major differences. The zDNA helix is left-handed and has a structure that repeats every other base pair. The major and minor grooves, unlike A and bDNA, show little difference in width. Formation of this structure is generally unfavorable, although certain conditions can promote it, such as alternating purine-pyrimidine sequence, negative DNA supercoiling, or high salt and some cations. zDNA can form a junction with bDNA in a structure which involves the extrusion of a base pair. The zDNA conformation has been difficult to study because it does not exist as a stable feature of the double helix. Instead, it is a transient structure that is occasionally induced by biological activity and then quickly disappears. It is possible to predict the likelihood of a DNA sequence forming a zDNA structure. An algorithm for predicting the propensity of DNA to flip from the B form to the Z form, SUNT, was written by P-Shing Ho in 1984 at MIT. This algorithm was later developed by Tracy Camp, P. Christoph Champ, Shandor Maurice, and Jeffrey M. Vargason for genome-wide mapping of zDNA.